A groundbreaking discovery has shed light on the early triggers of blood cancer, revealing a complex interplay between cellular components in the bone marrow. The key culprit? Inflammatory microenvironments that disrupt the delicate balance of blood and immune cell production. Every second, our bone marrow tirelessly produces millions of new blood and immune cells, a process that relies on harmonious cooperation between hematopoietic stem cells, HSCs, supportive stromal cells, and immune regulators. However, over time, this harmony can unravel. Aging, persistent inflammation, and somatic mutations can distort the communication between these cellular partners, leading to a silent yet dangerous expansion of mutated HSCs. This phenomenon, known as clonal hematopoiesis of indeterminate potential, CHIP, affects a significant portion of the elderly population, with roughly 10 to 20% of adults over 60 and nearly 30% over 80 being impacted. Despite being asymptomatic, CHIP carries a tenfold increased risk of blood cancers and doubles the chances of cardiovascular disease and premature death. Myelodysplastic syndrome, MDS, a related HSC clonal disorder, further complicates matters. It affects up to 20 in 100,000 people over 70, and a staggering 30% of these cases progress to acute myeloid leukemia, AML, an aggressive and often fatal blood cancer. The clinical importance of these disorders is undeniable, yet the role of the bone marrow microenvironment in their development has remained shrouded in mystery. To unravel this mystery, an international team, co-led by Judith Zorg from EMBL and the University of Basel, and Borgen Gezgez from UMC Mainz, embarked on an in-depth molecular and spatial analysis of human bone marrow. Collaborating with Uwe Platzbecker at the National Center for Tumor Diseases, NCT, Dresden, the team utilized single-cell RNA sequencing, biopsy imaging, proteomics, and functional co-culture models to create a high-resolution map of the bone marrow microenvironment across healthy donors, including those with CHIP, and patients with MDS. Their findings revealed a startling cellular transformation that occurs well before any clinical signs of disease. The researchers identified a population of inflammatory stromal cells that replace the normal, stem cell-supportive mesenchymal stromal cells, master of science. These inflammatory MSCs, or IMSCs, release large amounts of interferon-induced cytokines and chemokines, attracting and activating interferon-responsive T cells. This creates a self-perpetuating cycle of chronic inflammation, suppressing healthy blood formation and promoting vascular remodeling. But here's where it gets controversial. The scientists discovered that the mutated hematopoietic cells in MDS do not directly drive this inflammation. Using a new computational tool, SpliceUp, developed by Maxim Kolmatov, co-lead author, and EMBL alumnus, in collaboration with Pedro Maurer and Ivar Hellström Lindberg from Karolinska Institute, the team could distinguish mutated from non-mutated cells within single-cell data based on aberrant RNA splicing patterns. This distinction revealed that the inflammatory network in the microenvironment becomes dominant in MDS, overshadowing the bone marrow's regenerative architecture. Karen Prummel, co-lead author, and EMBL postdoc, highlighted the significance of this finding. Another striking observation was that MDS stem cells couldn't trigger stromal cells to produce CXCL12, an important signal that triggers blood cells to settle in the bone marrow. This failure may help explain why the bone marrow stops working properly. Maxim Kolmatov added, it was surprising to see the lack of a direct inflammatory effect from the mutant cells. However, when considered alongside changes in the T cell and stromal compartments, it emphasizes the critical role of the bone marrow microenvironment in disease progression. This discovery positions inflammation as a central player in the earliest stages of blood disease and identifies the bone marrow microenvironment as a crucial therapeutic target. By shifting focus from mutated stem cells to the cellular ecosystem that sustains them, researchers open up new avenues for preventive and therapeutic strategies. Anti-inflammatory or interferon-modulating agents may preserve bone marrow function in older adults with CHIP, while targeted drug combinations and microenvironment-directed therapies could halt progression to MDS or AML. The distinct molecular signatures of IMSCs and interferon-responsive T cells also offer potential biomarkers to identify individuals at risk long before clinical symptoms arise. Borhen Gezgez, principal investigator in the Department of Hematology at UMC Mainz, emphasized, "Our findings reveal that the bone marrow microenvironment actively shapes the earliest stages of malignant evolution." Understanding the interactions between stromal and immune cells provides a foundation for preventive therapies that intercept disease progression before leukemia develops. This work expands our understanding of inflammaging, the chronic, low-grade inflammation underlying many age-related disorders, including cancer, cardiovascular, and metabolic diseases. The bone marrow, once viewed solely as a blood production site, now emerges as both a target and a driver of systemic inflammatory aging. By uncovering the mechanisms of local immune-stromal interactions, this study provides a new framework for investigating inflammatory remodeling in other myeloid malignancies and advanced leukemia. Judith Zorg highlighted the importance of longitudinal studies. Our current findings are based on cross-sectional data, which has implications for therapies that replace malignant cells while leaving the bone marrow niche intact, such as blood stem cell transplantation. We are now investigating the extent to which the niche retains a memory of disease, which could influence its response to new, healthy stem cells. This groundbreaking research not only advances our understanding of blood cancer development, but also opens doors to potential preventive measures and targeted therapies. The future of blood cancer research looks promising, with these new insights guiding the way.